now know that we can either reverse or slow down the aging process.
Aging is going to be a crisis. By the year 2030, I think it is, there'll be 55 million of us over the age of 70.
What we'd really like to do is to extend the health span of individuals while they're alive. If we can maintain health, that's an important goal.
It's not a question of vanity. Everybody likes to look good. But at the same time, what counts is what is your true biologic age? Can you maintain vigor, and can you function at a level that you functioned at when you were in your 20s and your 30s or even younger?
I'm now approaching the feeling that I'm beginning a new life with new adventures and new excitements.
I don't think it's a matter of turning aging around. I think it's a matter of preserving what intrinsically is good in the body and healthy in the body, and slowing down maybe disease processes or processes that make muscles get weaker.
These tools, that we have now for genetics and genome efforts, are so powerful that even the genetics of something as complex as aging and longevity can now be approached, can now be investigated. That simply would have been unthinkable 5 years ago.
And I feel as though life is just beginning, just opening up. Possibilities are endless.
Age is only a number. Like it or not, we begin to age the moment we're born. However, given that our bodies are designed to live productive and vital lives well past the age of 100, 110, 115, this should not be an issue.
The limit so far has been a practical 122 years, strictly from that's how long the longest individual that we can document lived. There's no particular reason to believe that individuals can't live to at least 130 or possibly more.
As a population, we are living longer, slightly. In the last 100 years, if you correct for birth, deaths, let's say when children died very early in life, we have only extended the average lifespan in 100 years, 3 years and 7 months. With all of our technology, with all of our knowledge, how is it we're only living 3.7 years longer on average? Well, we're going to explore that.
The fact is that we haven't done anything about the basic processes that lead to aging. All we've done is reduce the infectious disease rate and deaths from infectious disease by good hygiene, and prolong the lives of people with certain acute illnesses by curing them with antibiotics. But those aren't the things that really lead to age-related mortality.
The idea was in the old days that people would develop up to adulthood, and then it was sort of just all downhill after that. I don't think that was ever true. If you look at the productivity of the people we admire most, the great writers, the great musicians, they were all doing their best work in their last years. Nevertheless, there was this feeling that there was simply nothing but misery after a certain age.
Most of your medical schools treat aging as gerontology, and gerontology is the finger in the dike. You make a diagnosis and you say, this guy has Alzheimer's. Or you make a diagnosis and says this person is suffering from congestive heart failure with enlargement of the heart, and so on and so on. But what do you do about preventing it? What we need to do is develop successful aging, which allows us to intervene to improve the quality of our later years.
I think what we're discovering about aging is that it isn't an inexorable process, that things don't just wear down, but there are genes that basically turn on programs that trigger aging. Believe it or not, there's some reason to believe that estrogen regulates some of those genes in terms of keeping the organism young. You've heard this research about telomeres, and that basically aging has to do with these telomeres. But there's good reason to believe that if we can control a certain number of genes and gene programs, that we'll be able to have, I don't know, immortality, but we'll be able to prolong life much more than we can today.
The bottom line, we begin to age the moment we're born. How long the process takes and how well we do depends almost entirely upon how well we take care of ourselves. The human body is a collection of cells. Some estimate as many as 100 trillion. But that's not the issue. The point is, no serious discussion of aging process can begin without first considering what is taking place at the molecular level as we grow older.
Human cells will replicate somewhere between 40 to 80 times, depending on the exact tissue. At that point, they simply stop. They, in a sense, they've aged, and they just stop replicating at that point. What happens to those cells, though, is not only do they not continue to divide and make new cells, they also start changing the kind of proteins they make. A growing fibroblast makes collagen and related materials that provide the matrix that cells grow on. When a cell becomes senescent, in other words, it reaches this point of no longer being able to replicate, it now stops making collagen and starts making enzymes and proteins that degrade the collagen and this matrix. That leads to disruption of the tissue.
I think that the fundamental deficit in aging is cell energy. I think aging is primarily a mitochondrial loss, the loss of your ability to burn energy, to maintain the repair and your body function requires the ability to utilize oxygen and to dissipate oxygen.
Obviously, oxygen is life-sustaining, but it has a downside. And the downside is that it causes destruction of many of the body's molecules within the cells and limits the lifespan of cells and organs and tissues and increases the risk for disease and so forth.
Modern theories of aging generally fall into two camps: damage theories and program theories. Now, damage theories are primarily concerned with the damage that occurs inside the cell over time. Program theories center around the idea that aging results in large part from a sort of genetic clock that decides when cells can no longer operate and reproduce at a rate sufficient enough to maintain optimal health. These distinctions are not necessarily mutually exclusive, but they do serve as a useful guide for discussing current thinking on how and why we age the way we do.
So there's a constant battle going on within the cell between the damage that occurs and the repair of that damage to return the cell to where it should be at homeostasis.
You know, through the normal process, these cells are aging, but they are replaced. If somehow some of these just stay around, you actually shift the balance so that there are more dysfunctional cells in the tissue. So I think both cell proliferation and cell death and maintenance of cells who live throughout the lifespan of a tissue are three different aspects of aging.
We need to have cell division in order to grow and in order to make structures, but too much cell division leads to cancer. So we need to learn how to regulate these two sides of the same process, in a sense. How do we maintain the ability of cells to replicate, but not in an uncontrolled way that leads to cancer or various kinds of hypertrophy? So regulating processes is very important in understanding aging, in my opinion.
The causes of premature aging in our society are bad dietary habits, use of alcohol, use of tobacco, obesity, not getting enough exercise, and all of those things are going to lead to deterioration of function before they would normally occur.
There are more heavy metals in our environment today. We have exhumed the bodies of people from centuries ago. We have 400, 500 times as much lead and cadmium and mercury in our bodies today as our ancestors did. And
what does that mean? If the people at home are thinking, all right, we're more toxic, we have heavy metals, what's that mean? What does that do to a person's biochemistry that can either increase the aging process or diseases associated with it?
Well, the heavy metals are really powerful impairment factors in the enzyme systems in the body. Our bodies are biochemical factories. Enzymes determine how rapidly hundreds and thousands of biochemical reactions go each day, and those heavy metals interfere with the essential minerals that are becoming more and more deficient in the American diet as just one manifestation. They cause free radical damage. They cause damage to DNA. so that our cells don't even reproduce in a healthy way. And this is a cumulative process so that the older we get, the more we have of these toxins.
You know, it isn't clear what role environmental pollutants play in the aging process. They're almost certainly important for carcinogenesis, and I wouldn't be at all surprised if the damage to cells from chemicals in the environment is a subtle cause of accelerated aging. But none of this has really been well documented, and the problem is that by the time we get around to documenting it, there'll be another 1,000 pollutants in the environment that we don't know anything about. I think the real answer is to have better controls on the emissions from our power plants and from chemical plants and so on, and just assume that putting a lot of chemicals with unknown effects into the environment is probably not good for life.
So in effect, the more mercury exposure someone has, as well as other metals, the less efficient their body becomes in normal metabolic process. And since Americans are so chronically fatigued, we're suffering from literally an epidemic of degenerative diseases that many physicians and scientists, public health advocates, believe that we've got to first recognize the problem and not have a political, buffer, right? We have to just be honest. If we've got a problem, what's the problem? The orthodox model, unfortunately, does not have a tool currently to take the toxins out outside of chelation therapy.
We've been using chelation therapy for 40 years now to help people with hardening of the arteries. But that's still controversial because we're fighting the multi-billion-dollar industry in operations, angiograms, and so forth. But there's no argument that heavy metals are removed by specific chelating agents, and we have several agents that we use, and it is dramatically effective, and yes, I think we are going to see that adopted.
Good. Well, I thank you for all the work that you've done in the field, because what at one time was considered preventative medicine, then was called holistic medicine, then alternative medicine, now complementary medicine, really is just anti-aging medicine, because whatever you stop from happening, you're extending the quality and quantity of a person's life.
We have actually started to look at what we would consider some preclinical type of risk factors, things that wouldn't normally be considered necessarily abnormal, but things such as increased arterial stiffness that we measure with a special probe that we place on the arteries, and also looking at the minor thickening of the walls of the carotid arteries by means of ultrasound. We think that these are markers of preclinical disease, and that perhaps if we can detect this and see what the risk factors for the development of these early markers are, that we could then intervene at an earlier stage and actually be more successful at preventing the development of overt cardiovascular disease.
There's some outstanding work being done in the science of gerontology and genetics, trying to understand what's causing the cell to speed up its aging and what can be done to slow it down, and where there's damage to repair that damage. Let's go on a little tour now and see from Johns Hopkins to Harvard, from MIT to Tufts and other places, that you'll see some of the cutting edge information that could mean the difference of you living to be 130, 140 with that extra vitality and without the fear of disease.
My lab has been studying aging for about 9 years now. We're interested in figuring out the molecular mechanisms that cause aging. We're studying a particular aspect of chromosome changes that we've learned about from yeast that are a part of what causes yeast cells to age. And we know in yeast we can intervene genetically in this process and slow down aging. We chose yeast for basically two reasons. One is that yeast cells are very similar in the way they do business to mammalian cells, including human cells. And the second is we can make progress rapidly in the yeast system. And we've in fact learned a lot about the aging process in yeast cells. So we're interested in figuring out what exactly controls this process and what's happening in these aging cells. And to show this with a picture, here are 3 young yeast cells and 2 old cells. And in this picture, we're looking at the nucleus of the cells. And the nucleus is where the chromosomes lie. In these young cells, the blue stain is highlighting the chromosomes of these cells, and the red is highlighting a part of the nucleus that's called the nucleolus. And what happens, as you can see by comparing these 3 young cells to these old cells, is there's a gross distortion in the nucleolus in the aging cells. And this chromosome instability in the nucleolus is what we think is an important component of the aging process, and in fact is a cause of aging in these cells. So it's important in aging studies to distinguish between correlations, that is to say, things that happen as a consequence of aging, and causes, that is to say, things that actually trigger the process. And one way to think about the difference between correlations and causes is if one could intervene to affect a cause of aging, then one could actually slow down the aging process itself. Whereas if one intervened to affect a consequence, for example, skin changes in people, that would have cosmetic benefit, certainly, but it wouldn't affect the underlying causes of aging, and we would still grow old at the same rate and have the same lifespan. So what you want to do is identify mutations, that is to say, changes in single genes in the organism that either slow down or speed up the aging process. And of course, it's really better if you can identify mutations that slow it down. And those are really the gold standard of aging mutants, is those which have a longer lifespan than the wild type. Now, if you can obtain such mutants and you have a screen that works, then you're in a position to identify what the genes are by standard genetics, which includes cloning, and identify the sequence of the gene, and to try and figure out what the protein product of the gene does. And that leads you to the process that is relevant to the aging in that particular system. Now, stem cell research is yet another approach in aging that's right now very exciting, because for the first time it's possible to culture these stem cells and to manipulate them in a petri dish. And the important thing about these cells is that they're pluripotent, that is to say, they're capable of differentiating into all the different kinds of cells in our bodies: neurons, skin, blood, muscle, and so on and so forth. So it raises the possibility of being able to manipulate these cells in a petri dish as stem cells with their potential to develop into other kinds of cells, and injecting them in situations where cell degeneration is causing problems, for example, neurological damage in Parkinson's disease. And the question is, and the promise is, can one inject stem cells, introduce stem cells in these individuals, have them differentiate into the appropriate neurons in the case of Parkinson's, and actually correct the defect in these people? Or possibly to have them differentiate in a petri dish, perhaps into an organ system, so that they might be of use in organ replacement. I think there's a tremendous potential for using these cells, which is being explored, but we're just at the very infancy of stem cell research right now.
For a long time, it's been known that there are tissues in the body which are always renewing themselves, like the blood cells. For these, there are stem cells that are located in specific places. For marrow and blood and white cells, it's in the bones, the marrow of the bones, and the thymus in young children. It was thought, however, that for many tissues, they're composed of cells which are terminal cells, which never divide again, and that those tissues therefore have no renewal capacity. There are two ways that thinking has begun to slip. One way is that there are now stem cells that have been discovered that remain in brain and some other tissues. How to wake them up and whether they could really replace cells that are being lost is what's now being investigated. The other way is to go back to early embryological development, where everything starts from one cell, and clearly you can then make new neurons, new kidney cells. So the question is how much you can understand about early development, and could you then use that to remedy and rejuvenate tissues or cells that are being lost in old age. There are other indications that this kind of permanent terminal state of cells is not as permanent as we once thought. The famous case of the cloning of Dolly the sheep and of the cloning of mice show that the nucleus that's been in a terminal cell can be put back into an egg and start all over again. That was a big surprise. It's a big change in people's thinking. And again, it's part of the notion that maybe it'll be possible to start again and make new cells to remedy the loss of cells during the aging process. Many groups have been working on a process called telomere shortening as a feature of aging. And for example, we expect that in these cells, telomerase, the enzyme that can promote telomere maintenance, will be shut down. What are the other genes? This complete genome approach, where you compare all the genes that are being expressed in the mortal cell and the immortal cell, gives you all the candidate genes that may be regulating or involved in the process. Again, that's very much work that's ongoing. There's already been a set of genes, an initial set of candidate genes that have been isolated, and the work is beginning to see whether those are the critical ones. If we could understand how cells make the transition from non-growth to growth and could control it, we might have a better idea of how to get stem cells to kick in and give us replacement cells. I think that if we think about longevity itself or any of the processes that are beginning to fail or cause trouble with aging, there is a genetic component. There's also an environmental component. The environmental component is the most important. For example, the Institute has turned out a wonderful manual of exercise in the last year and a half, which is having some impact and could have more. There's no question that individuals who maintain a reasonable exercise schedule can also maintain a very large fraction of their muscle mass and strength to very advanced ages. That's something that's under their control, just as smoking cessation could be under their control. These environmental factors clearly could contribute to much better health and quality of life as people age. The genetic factors are just beginning to be known, and it's not clear, but we may be able to intervene with them. For example, in the last years, brilliant work of a number of groups has begun to discover some of the genes that are involved in the development of Alzheimer's disease, in particular in the development of the tangles in the brain that tend to then block or wreck neuronal function. Based on that, several large pharmaceutical companies now have drugs that are being tested which could help to prevent the development of the tangles. Therefore, knowledge of the genetics, the genetic factors involved in the etiology of Alzheimer's, is already producing potential drugs that could help to prevent the condition. And that's one of a number of cases that we're hoping to augment in the coming years. The field is very technology-driven. Molecular biology has always waited for the next big technique. But the kinds of techniques that are being used are the modern molecular biology techniques in which usually tiny amounts of genes and materials are being manipulated and looked at. The impact of genetics and a Human Genome Project is enormous, and it's accelerating. For example, let's suppose that we want to find a few genes that are involved in asthma. That's not particularly age-related. We could do the same thing for diabetes. One of the ways to do this is to go to a very large family in which asthma is present in many of the individuals and study the genetics of that family. See, people used to think about doing something by curing, helping the memory of people who have Alzheimer's. The entire field is shifting, trying to use genetics to prevent the development of these age-related conditions and disorders. That's a big change, brought about in part by the Genome Project, the availability of modern genetics, knowledge of many of the genes. In fact, we're coming up to a knowledge of the full catalog of human genes in the coming 2 or 3 years. But the complex traits, the complex diseases of old age, diabetes, Alzheimer's, osteoporosis, and many conditions like loss of muscle mass, have many genes that can contribute small amounts. And in individual families or populations or persons, the specific change in one of those genes combined with the environment of that person, their diet, their habits, can tip the balance to cause the disorder. In other individuals, that gene change might have no effect, but a change in another gene might be critical. Or in the same individual on a different diet, that form of the gene might have no effect. So these are very complex questions. Nevertheless, when you can define one or two, a few of the many genes that can be involved in causing the disease, you can then use modern molecular biology and genetic techniques to find the others. And gradually you get to know much more about the pathway that's involved and to see places where you might intervene with a drug or a dietary change or a change in personal habits that might make a big difference in avoiding the onset of the disease. The other way of looking at aging is to wonder whether there's an underlying process independent of any specific disease or pathology. People have different opinions about whether there is that underlying process, but we know that we can attack each of the specific diseases that are associated with aging. And by doing something about them, we can improve the quality of life and have a real effect at all ages, not just thinking about longevity.
I certainly hope that the geneticists are able to come up with programs that can help stop the diseases that ravage us. If you came up with a gene that could help a person with cystic fibrosis or Alzheimer's, we're going to support that. But I do not believe that in the near future they will have that elusive magic gene that will give you an extra 20 years onto your lifespan.